I'll talk about our mouse work, which is and emphasise the behavioural studies that we're doing to fit, as the other speakers have done, with the main theme of the symposium. So when we look at human behaviour, be it healthy behaviour or behaviour which is involved in psychopathology, we can basically narrow it down to the way that we respond to punishing or negative stimuli in our environment, and the way that we respond to rewarding stimuli. And what we're looking at here is focusing firstly on our responses to punishment, looking at the way that we respond healthily to punishing stimuli. So, for example, an aversive stimuli could be upsetting our boss at work. Okay, we know that this is a very strong emotional stimulus. You don't want to upset the boss. You could be involved in a work task, so a student taking an exam or some writing a grant, and you want to succeed, you want to avoid the failure. And in order to do that, you need to be motivated, and that requires effort. And what you will experience if you successfully escape or avoid the punishment is an emotion of relief. And what you're trying to avoid is this emotion of fear or helplessness. Now, if we look at the pathology of that behaviour, this could be in depression or depressive episodes of bipolar disorder, or in schizophrenia, for example, then there will be two basic pathologies in this responsiveness to punishment. There will be firstly a reduction in effortful motivation, so it will just feel like effort, and there will be a reduction at the emotional level, so the experience of relief will be reduced, and the emotion of fear or helplessness will be increased. Now, if we want to study that in mice, then we need tests which allow us to measure both effortful motivation and these emotional responses of relief, fear and helplessness. And there are such tests which exist, which I will talk about. For example, if we look at fear conditioning, this is the learning of an association between a conditioned stimulus, such as a tone, and an unconditioned punisher, such as an electroshock. This will tell us about this type of emotion, but it won't tell us anything about the effort involved, because here all that the animal can learn and can do is freezing behaviour. This doesn't require particular effort. So it's what we call a P+, E- behavioural test. Another type of test would be the two-way escape or avoidance test, which is also referred to as helplessness. So you can pre-expose the animal to an inescapable punisher in the form of electroshock, and then subsequently measure its ability to learn that it can now escape or avoid this electroshock. And this will require physical effort, and it will require some cognitive effort to learn the association which now exists between behaviour and the effect of that behaviour on the environment. Another example would be a treadmill, where the animal needs to make physical effort in the form of running in order to avoid an electroshock. So there are mouse tests available which allow us to study these various components of punishment-related behaviour. In collaboration with TSE, which is here in Poland, in association with another company, which has the part of the exhibit, we developed several years ago a system that TSE calls the multi-conditioning system, which within one system allows us to measure both two-way escape and avoidance and freezing behaviour, so that we can combine relief, fear and effortful motivation with one behavioural test system. This is an example of using that multi-conditioning system to develop a test which you might call a test of helplessness, learned helplessness. We prefer to be more sort of more parsimonious and call it a test of learned aversive uncontrollability. And what this involves is pre-exposing mice to either escapable or controllable shock or uncontrollable shock, and then looking at the effects of that controllability or uncontrollability on subsequent responsiveness to a now controllable electroshock, a controllable punisher. And what you can see here, if we do this, these are the data from the two groups of mice at the second pre-exposure, comparing the escape behaviour of the mice that are getting the escapable shock, the controllable shock, and the transfers from one side to the other of the mice that are getting exposed to the inescapable shock. Now these attempted transfer are having no effect on the electroshock. And what you see already after the second pre-exposure is that those mice getting exposed to the inescapable shock are starting to make less transfers. Now what's also important to know here is that the amount of shock that these two groups receive is identical, because the escape response in the ES mouse determines the duration of the electroshock in the mice exposed to inescapable stress. In other words, the only difference in the experience of these two mice is the controllability of the electroshock. And when we give the animals an escape test on day 6, we see that the mice exposed to escapable or controllable shock are demonstrating escape responses, and those exposed to the inescapable shock are showing a much lower level of escape responses. And this effect persists across days, but these mice will eventually learn to escape. Now, this is not in our opinion, is not a test, is not a model of depression. What it is a very important demonstration that mice have a concept of controllability and uncontrollability. And this, of course, is very important if we're going to talk about using animals to study these pathologies of human behaviour, such as helplessness, feelings of loss of control. Just compare this, if you would, with the forced swim test. In the forced swim test, the animal would be placed in water and it gradually starts to float. And there are, of course, many explanations for this. Some people call it despair or despair-like, or helplessness-like. The important point is we have really no idea what is causing this behaviour, because there is no control group. In this test, we know that what's important is uncontrollability, because we have a group exposed to controllable and a group exposed to uncontrollable, and we can demonstrate the effects of the uncontrollability. So that then is an example of the sort of test one can do to look objectively at mouse responses to punishers. Now, what about responses to reward and the pathology of responses to reward? Well, if we're presented with rewarding stimuli, which very often will require effortful motivation to obtain those stimuli, our pleasure experience will be high, our feelings of sadness in making effort to obtain those stimuli will be relatively low. In a pathological condition, so somebody that, for example, a patient with depression who's showing a reduced interest in those things which are normally rewarding, then there will be a deficit at the emotional level. The responsiveness, the sensitivity to pleasure will be reduced, feelings of sadness will be increased, and alongside that, again, there will be a deficit in the effort made to obtain those pleasurable stimuli. How can we go about looking at that in animals? Well, one test which can be used to look at the interest in the stimulus, the liking of the stimulus, if you like, will be the two-bottle sucrose versus water test, which is a test of, certainly of reward sensitivity, but of course requires very little effort. If we want to look at effort to obtain gustatory reward, then we need to use operant tests, such as, for example, a progressive ratio schedule, where the mouse needs to make an ever-increasing number of operant responses to obtain successive rewards. And of course, as well as gustatory stimuli, social stimuli are also important. This is an example of a social motivation test, social approach. Of course, it has a rewarding component, but it doesn't have an effortful component. It's possible to train mice under operant conditions, so using a nose poking response or a bar pressing response. For example, this is showing behaviour in a spatial reversal test, where the animal is presented with two nose poke stimuli, one of which responding to one of which will result in reward. Responding to the other one will result in in no reward and a timeout. And you can switch, you can reverse the relationship between these two stimuli and which one obtains reward. And the mice will show stable behaviour across weeks in this test, allowing you to look at environmental challenges, pharmacological challenges. So what we end up with then are a number of tests which allow one to look at responses to rewards, responses to punishment, some of which also require one to look at this very important variable of the effort required to escape or avoid the punisher or to obtain the reward, and those are relevant to the research domain criteria, domains of positive valence, negative valence, and some of the other domains that have been proposed. And what's important about this domain approach is that it goes across diseases. So across a number of diseases, we have patients that show apathy. So it's important to understand what's causing that apathy. It doesn't have to be specific to a certain disease. We have patients which show helplessness or describe feelings of lack of control of their environment across disorders. It's important that we have models for helplessness. Now, in order to induce the pathologies in these tests, we use a number of approaches. The one I'll talk about this morning is chronic psychosocial stress, where we expose the black 6 mice that we're interested in to a continuous exposure to aggressive CD1 mice, and the two mice are placed together for 1 minute of attack per day, and the rest of the time they are in continuous visual, olfactory and auditory contact with this dominant aggressive mouse. And what we can see now by combining this manipulation with these tests is now we can start to talk about some interesting psychopathological relevant effects. I said before that this two-way helplessness test provides us with nothing more than a measure of uncontrollability, learned to an electroshock. But what we see now, if we look at mice that have been exposed to chronic social defeat and compare those with control mice, is that the mice that have experienced social defeat demonstrate spontaneously a deficit in avoiding and escaping the electroshock in this two-way active avoidance test. Okay? So they have never experienced this electroshock as uncontrollable, but they have experienced a lack of control in a social situation, and they appear to generalize that into this avoid escape test. So we have what we could call a state of generalized uncontrollability, or a state of generalized helplessness, now in a model which involves a punishment component and an effortful component. So if we look in detail at these mice, they have trouble they show a motivational deficit in responding to the electroshock, and they also show a deficit in learning that this stimulus is avoidable or escapable. What we can also show, using the same apparatus, is that they show an increase in fear conditioning. So at the purely emotional level of how emotionally relevant, how emotionally how fearful are they of this stimulus, we can see that the chronic social defeat in a social context increases the fear learning about a physical, painful stimulus. So again, a generalization between different punishing stimuli. If we place the animals on a treadmill, which they have to run on in order to avoid the electroshock, we again see that the chronic social defeat reduces a deficit in the effortful motivation to escape or avoid this physical punisher. Here we're looking at where the treadmill, as it's running at a speed of 20 centimeters per second, the majority of the chronic social defeat mice are able to run on the treadmill at this speed and are comparable to the control mice in terms of the number of shocks that they receive. But when we increase the treadmill speed to a running speed, we can see that we can really bring apart the stress mice and the control mice, so that now the running time of the chronic social defeat mice is markedly reduced compared to the control mice. So what we're doing here is that we're saying that the maximum amount of shock that the mice can receive is 10 seconds, and the maximum test duration is 1200 seconds, and we see that the control mice are all able to run for 1200 seconds for 20 minutes without getting to this 10 seconds of electroshock, whereas the majority of the stressed mice, the socially stressed mice, receive the 10 seconds of shock within this 20-minute period. So it's also affecting the motivational effort to avoid or escape from a punisher in this under these conditions of physical effort. If we look at the effects of psychosocial stress in a reward test, which does not require effort, so in the two-bottle test, we see that there is no effect of the chronic social defeat on the drinking, the absolute drinking of saccharin. They consume a bit more water, and there is no real effect in the preference for saccharin. They continue under these conditions of low effort to have a marked preference for saccharin. If, however, we test the mice under effortful conditions, now for sucrose pellets, in a progressive ratio schedule operant test, where the mouse is a very simple test in terms of what's required at the level of learning. The mouse simply has to make nose pokes in order to obtain the sucrose pellet, but the number of nose pokes it has to make, the number of operant responses it has to make, is increasing progressively on a ratio scale. And if we look at the final ratio that the animals achieve, and therefore the number of pellets that they earn, we see that that is reduced in the psychosocially stressed mice compared to the control mice. So if we make the animals work for reward under effortful conditions, then we see the effects of the psychosocial stress going in the direction of an apathy in these mice, showing a deficit to obtain reward under effortful conditions. We can also look at chronic social defeat in the IntelliCage, where you divide the IntelliCage into two. On one side you have your control mouse or stress mouse. On the other side you have the stressor, the aggressive CD1 mouse, and then you can continuously monitor the effects of the psychosocial stress on the animal's effortful behaviour to obtain saccharin in the home cage or water in the home cage. And here too we see that the psychosocial stress induces an apathy type effect, in that the animals over time, so here in 5-day blocks, here baseline, and then the first 5 days of chronic social defeat up to the last 5 days on this 15-day schedule, and we see then the chronic social defeat mice during the active period, when the animals are showing a lot of operant behaviour for saccharin as well as for water, we start to see deficits in the chronic social defeat mice in this effortful home cage environment. In terms of the neurobiology underlying this is of course why we're doing this, because it will give us objective models with which we can then start to look at the neurobiological basis of helplessness and apathy. What we can see is that with, for example, with a lesion, a depletion of dopamine, specifically in the nucleus accumbens, we can induce a very similar effect in the progressive ratio task to that which we see in the chronic socially stressed mice. So as I said, with chronic social stress you get a decrease in operant behaviour on a progressive ratio schedule to obtain reward, and you see a similar deficit in mice that have been exposed to 6-hydroxydopamine in the nucleus accumbens to deplete the dopamine function in the accumbens. Using the same manipulation, so a dopamine depletion in the accumbens, you can recreate this stress-induced deficit on the treadmill. So here we're seeing comparing 6-hydroxydopamine mice in the accumbens with vehicle mice, and we get a very similar effect in terms of this deficit in the effort to escape or avoid the electroshock that we see with the chronic psychosocial stress, providing some indirect evidence that one of the regions affected by chronic psychosocial stress is dopamine function in the nucleus accumbens. And so we're using various techniques now to using this model of psychosocial stress with the behavioural tests that we've developed, using this to investigate this circuitry. This is what we're proposing to be the normal adaptive state of the control mice, and these some of the changes that we're proposing and that we're investigating in the social stress mice. So focusing on the amygdala in terms of the emotional processing of the reward or punisher in terms of its emotional significance, and focusing on the VTA and its outputs to the accumbens and to the prefrontal cortex in terms of the effortful motivational deficits that we see in these mice. And I'd like to thank my group, Hannes Siegrist, who's here, who's performed a lot of these experiments, also the funding bodies, and also like to thank TSE for their collaboration in developing some of these apparatus, and the organisers and yourselves. Thank you very much.